Well, 9:03 seems like a good time to start. All right, let's do it. Okay, welcome everyone. This is the first CART Distinguished Lecture of the year. As most of you know, our Distinguished Lecture Series is a once-a-month CART program that goes from October through June. And this year, as every year, we have a great series, excellent guest speakers lined up to provide you with the latest scientific updates on autism spectrum disorder. Just wanted to thank a few folks before we get going today with our speakers. So the UCLA CART Distinguished Lecture is sponsored by the UCLA Targent Center, the UCLA Intellectual and Developmental Disabilities Research Center, the UCLA Brain Research Institute, the UCLA CAN REACH training program. Anyone else? I think that's it. Okay. Attendees on Zoom can ask questions through the lecture using the Q&A window. And we'll also take questions from the audience, both Zoom and in-person audience at the end of the talk. And we'll be recording this talk and it'll be posted to our website with all the other talks from CART. So it is my great pleasure to introduce Dr. Carrie Bearden today. And she, of course, needs no introduction, but I'm just going to go ahead and introduce her because there's so many fun things to say. So Dr. Bearden, as you know, is a professor of psychiatry here at UCLA and in psychology as well. She received a PhD in clinical psychology from the University of Pennsylvania and completed clinical training in San Diego. Joined the UCLA faculty in 2003. Her work aims to understand neurobiological risk factors for the development of severe mental illness in youth, both in clinically defined high-risk cohorts and in highly penetrant genetic conditions. She's particularly well known for her research in taking a genetics-first approach to studying brain mechanisms underlying the development of severe mental illnesses. She's the director of the UCLA Center for Assessment and Prevention of Prodromal States, the CAPS Center, which is a clinical research program for youth at high risk for psychosis. She also directs our UCLA neurogenetics training program. She has 350, I think is probably closer to 500 peer-reviewed by now, since we last checked. A lot of publications, is one of the world's most highly cited scientists. And she currently serves as an editor for the journal Biological Psychiatry, is the chair of the DSM-5 Serious Mental Disorders Committee, and is president of the Society of Biological Psychiatry. We're not going to list all the awards because that would take the whole hour. So she's a fellow of the Association for Psychological Science, the American College of Neuropsychopharmacology, many others. Dr. Bearden has received many awards, both for research and for teaching and mentorship, including the ACMP Joel Elks Research Award, the Biological Psychiatry Gold Medal Award, and the NIH Method to Extend Research in Time and Merit Award. And for those of you who are unfamiliar with the Merit Award, that's just about the best thing you can get from NIH. So that is a big deal. So that is very exciting. And just, and you all know this, but Carrie is such a passionate supporter of women in science and science in general. She's one of our biggest advocates and, you know, one of our most sort of active in the media, kind of championing all of our scientific endeavors at UCLA, especially during these really difficult past few months. She's been a real inspiration for those of us trying to hang on in the midst of all of this turmoil. So please join me in welcoming Dr. Carrie Bearden. We're so happy to have you.
Thank you so much for that awesome introduction, Lucina. I really, really appreciate it. It's such an honor to be invited to speak here for the first CART of the quarter, of the fall quarter, and to talk to all of you, and hello folks on Zoom as well. So on the note of the last point that Lucina made, I just wanted to point out that while I'm super excited to tell you all about science, this is also a really rough time for science and public health. And so, I think homework for all of us, including myself this weekend, go out and talk to everyone you know, your friends, family, neighbors, about what you do, about the importance of science and clinical care for kids with autism, whatever, if you do work in a lab, if you do clinical care, please talk to people about what you do, why it's important, and, why you need to be able to keep doing it. So thank you. That's my PSA. Moving on to my very most important slide of the entire talk. This is my thank you slide. And so I put it in the beginning because I do not want to shortchange all these fabulous people in the lab, both alumni and present members who have done all the incredible work that I just get to show up and tell you about. So you'll be seeing their lovely photos throughout the talk. And then I also want to thank all of our amazing international collaborators, including right here in this building, who have made all this work possible. So we have made some really, really incredible progress in our understanding of the genetic risk architecture of developmental neuropsychiatric disorders over the past decade. And so I don't think I have a pointer, but what we're looking at here is just the odds ratio for schizophrenia. Down there, the little blue dots on the bottom are for what we call common variants. Those are small effect alleles, SNP effects that single nucleotide polymorphisms that each of which individually confers a very small increase in risk for the illness. But you can see up on the other end of the odds ratio spectrum, copy number variants, those are deletions, that are duplications of DNA segments, as well as some single gene disorders that confer very high risk for the illness. And we have a similar plot down here at the bottom for autism, where you can see, and then this here is zoomed out of the high risk variants, so both de novo damaging variants as well as copy number variants that confer greatly increased risk for illness compared to common variants. And so the fact that these copy number variants and single genes are strongly associated with risk gives us an opportunity to leverage this what we call a genetics first approach for understanding the biology underlying developmental neuropsychiatric disorders. And the fact that you can model the same genetic defect in an animal really allows the development of animal models a strong construct validity, as opposed to, just focusing on a behavior and saying, your mouse looks depressed because they're not really swimming very much. And so I think this is a strong model, and really in that sense provides an ideal system in which to dissect circuitry mediating behaviors. From a perspective of human studies, can also be identified very early in development, sometimes even in utero, before clinical symptoms emerge. And so the particular one of these copy number variants that I'm going to be focusing in here today is at the 22q11.2 locus. I'll tell you a lot more about that. And so again, taking this genetics first approach, if you're taking a behaviorally defined clinical population or idiopathic population, I'm sure you're all aware there is an incredible amount of heterogeneity, which makes this very complex. And so at the level of a biomarker signal, you're again going to get a fair amount of complexity. Now, if you take a subset based on a shared and well-defined genetic etiology, you can get traction based on this increased homogeneity of your biomarker signal, which can then be studied at the level of neural circuits, systems, cognition, and behavior, which then in turn lead to these downstream symptom presentations. So while the majority of cases of developmental neuropsychiatric disorders remain of unknown etiology, these rare variants, and so here again, I'm speaking specifically about copy number variants, may account for a larger proportion of cases than previously believed. About 2%, we think, of schizophrenia cases, up to 25%, maybe even more, of cases of simplex autism spectrum disorder. But the fact that we have this phenomenon where we have one disorder that can be caused by, or at least caused is a complicated term, but can at least be associated with risk based on multiple different mutations, as well as the converse, one mutation leading to multiple different disorders, really provides some strong evidence for genetic pleiotropy, and also challenges these widely held views that we have of neatly defined diagnostic categories. So that's going to be a theme. So to tell you a little bit more about the 22q 11.2 microdeletion syndrome, it is also known as villocardiofacial or DeGeorge syndrome. It has an estimated incidence of 1 in 4,000 live births, 1 in 3 to 4,000. So it is a rare disorder, but not as rare as you might think. It is the most common contiguous gene deletion syndrome. It's a fairly large chunk of DNA that's missing on one copy of chromosome 22q. There are some associated physical features, cardiac defects, immune deficiency, craniofacial anomalies. You can see some of the folks who have this deletion here. We got really interested in it because of the very high associated risk of schizophrenia, about 20 times that of the general population. It's believed to account for about 1% of sporadic schizophrenia cases. There is, and this is just a plot from the C&V working group of the Psychiatric Genomics Consortium, just showing that genome-wide, it has the strongest association with schizophrenia. That being said, there is a lot of diagnostic pluripotency, elevated rates of autism spectrum disorder ranging from 12 to 50%. So a lot of variability across studies, as well as attention deficit hyperactivity disorder and anxiety disorder. I want to mention here that I actually got, for the trainees who may be listening, I got into this quite by accident. I was doing a clinical rotation at Children's Hospital of Philadelphia and just started seeing a lot of these kids for neuropsych evaluations. And I said, this is really, really interesting. Somebody should study this. And that's, that's how it all went. So some of the key questions that we've been really interested in over time, just first of all, as a starting point, we have limited data on individuals carrying the same highly penetrant copy number variant. So are there consistent and reproducible effects of the 22q deletion on brain and behavior across species? And secondly, are there scalable brain biomarkers relevant to sleep, cognition, and symptoms? And so sleep is a new part of the study that I'll be talking about a little bit. Then, I think this is a really critical question if we want to think about this as a model for idiopathic or behaviorally defined psychiatric disorders, is whether there's overlap between the genetic and neurobiological mechanisms underlying clinical phenotype in a highly penetrant copy number variant and an idiopathic illness. And then we're also interested in whether gene dosage effects, so whether there's a reduced or increased expression of genes within the locus, can that provide us clues into disease biology? And then lastly, are there distinct developmental trajectories that may be associated with different neurobehavioral outcomes? And here is my reminder to shout out to CART because when I came here, this was actually, I got a pilot study from CART in 2008. I checked the date, and that was a pilot study that allowed us to get started with imaging of this population here. And so that was actually the ground floor of that R37 grant that Lucina mentioned. That's been a prospective longitudinal study going on for 15 years now. So yeah, so lots to talk about. So we know that brain activity is organized, and actually Lucina has done some of the really amazing work on this. Oh, and I don't know if I can move this little screen sharing icon out of the way here so you can see my title. Yeah. So we know that brain activity is organized into large-scale functional networks, and this can be measured and mapped in a variety of ways, but one is using resting state functional MRI, which is a measure of spontaneous fluctuations in neural activity when one isn't engaged in any particular cognitive task. And there's a lot of support from biophysical models and other evidence really supporting this notion of functional networks within the brain that are pretty reliable and can be mapped across species as well. So there's emerging evidence. It really leads us to think of developmental neuropsychiatric disorders like autism and schizophrenia as connectopathies, as if you will, really disorders of interareal communication that are affecting large-scale neural systems. And this, so this applies certainly to the 22q deletion, where this is a prior study where we found pretty widespread patterns of hypoconnectivity in adolescents with 22q deletion in these large-scale brain networks. And these patterns of hypoconnectivity were so consistent and reliable that we were able to classify 22q deletion cases versus controls with 87% accuracy and actually generalized to a totally independent cohort. And actually within our 22q deletion patients, we were able to classify based on network patterns within a few networks, particularly the anterior cingulate cortex and the precuneus, the default mode network and the left executive, and a supplementary motor network. We were able to classify those that were at high versus low risk of psychosis based on these functional network connectivity patterns. Now, of course, this does not tell us anything about the underlying mechanisms. And so that was something that we were really interested in studying. And so this was a study that we did with my colleague, Alessandro Gozzi, and a wonderful postdoc in his lab, Elena Alvino, where we attempted to do a fully translational study to examine this functional connectopathy. And as I mentioned, our prior study was in adolescents and young adults. So we're really interested in developmentally how this manifests. And so in Alessandro's lab, they took juvenile and adult mice, and we tried to do the exact same methods in humans. This was a larger study where we had 139 deletion patients, 117 matched typically developing controls. And so this was a fun challenge to do things methodologically identically in humans and mice. I think we did an okay job. So we looked at both, we did a spatially unbiased global functional MRI connectivity mapping in the two groups, controlling for sex, number of retained volumes based on motion correction, and then a correction for multiple comparisons. And we were really focused on a age group by genotype interaction. And then we followed this up. We did some seed-based connectivity mapping as well, using the same methods. And so what we found, and here, this was collaboratively done by Charlie Schleifer in my lab, an MD PhD student, and Sylvia Gini in Alessandro's lab. And what was really interesting was we saw this actual developmental reconfiguration where we saw this pattern of pretty widespread hyperconnectivity in children with the deletion, which then in our post-pubertal group actually reverted to this pattern of hypoconnectivity within these networks. And so the bottom is just showing you that shift, looking at an age by genotype, showing you specifically the clusters that are showing this age by genotype interaction. And then with the seed-based approach, we actually saw the similar switch within the default mode and hippocampal networks. So Elena did the work in the mice, and this was a whole brain voxel-wise mapping of global fMRI connectivity in this large deletion mouse model. It's on chromosome 16 in the mice, really the syntenic region. And we were actually pretty surprised to see a really quite identical pattern to what we saw in the humans with this. The orange color is showing you the global overall hyperconnectivity in the juvenile mice, and then reverting to this more focal pattern of hypoconnectivity in the adults. And again, here's the individual data points, just so you can see that that age by genotype interaction. So then again, a similar developmental switch within the default mode and hippocampal networks. So in the mice, we can address some questions that are tricky to do in humans. So we're really interested in whether synaptic pathology may be preventing proper circuit formation during development, and is that responsible for this altered connectivity? And so what we're looking at here, this is the again, the fMRI connectivity switches that I showed you. And then that was actually mirrored by changes in spine density. And this is the top is the prefrontal cortex, and here is the hippocampus. And so what you can see is that there was an increased synaptic spine density in the juveniles, which then reverted to actually no differences in the prefrontal cortex between the wild type mice and the large deletion mice. But in the hippocampus, there's actually reduced spine density in the adult mice. So then, of course, that's just correlational. So we wanted to next address whether we might be able to get at whether there may be a causal connection between synaptic alterations and this observed fMRI disconnectivity. And so here again, we could really take advantage of what's known about the biology of the syndrome. And so there was a similar mouse model to the large deletion mice, not identical, but a slightly different mouse model that was studied in Joe Gogos's lab, where they found that there's a particular pathway, the GSQ3 beta pathway, that's overactive in this mouse model. And this hyperactivity in this pathway could actually be normalized with selective antagonist. And this was work done in Josh Gordon's lab. And so this normalization, we wanted to see whether, first, does this GSK3 beta normalization, does it rescue both the dendritic spine density phenotype in juveniles in the large deletion mouse model. And similarly, does it also rescue these fMRI alterations? And so and when I say we, I mean Alessandro's lab, they inhibited the GSK3 beta with the selective antagonist during postnatal weeks 1 to 3. And then they measured the dendritic spine density again in the prefrontal cortex and hippocampus, as well as whole brain connectivity. And what do we find? So interestingly, with the, as hypothesized, with this GSK3 beta inhibitor on board, we do see this, inhibition of, or essentially this normalization of of both the increase in spine density as well as the fMRI connectivity. And so, pharmacological inhibition of GSK3 beta normalized both, which was an interesting finding. And I should mention there, that was just in the juveniles. They did the same experiment in the adults, and they actually did not see changes. So this seems to be really affecting just as a developmental pathway. So are synaptic mechanisms implicated in human dysconnectivity? For this, we had to use a different method because we, can't do the same study in the same kind of experiment in humans. And so this was a method we are calling it gene decoding, but essentially we're looking at spatial correlations of gene expression in the brain with our age by genotype interaction map. So where is gene expression correlated with these particular, what genes are correlated with this map of our age by genotype interaction. And of course, you have to apply a really rigorous multiple comparison correction, correcting for spatial autocorrelation and so forth. But doing all that, I'm happy to talk later about methods. We do see, so we found there were a little under 4,000 genes that were significantly overlapping with these brain regions. And then we looked at whether these changes are occurring in brain regions that are spatially enriched for proteins that are interacting with GSK3 beta. And what we found is that when we look at GSK3 beta interactors broadly, we do see some enrichment, but it's pretty weak. Whereas if we focus in on synaptic GSK3 beta interactors, and this is based on the curated SynGO database, I'm looking specifically at synaptic interactors, we do see very strong enrichment. So again, not proving causality, but certainly suggesting that synaptic mechanisms are pretty relevant here. And then looking at just looking at the cell type specificity, we looked here at brain regions exhibiting the functional remodeling, that is that developmental switch that we saw. And we do find that they are spatially enriched for specific, specific neuronal cell types and synaptic mechanisms. So I don't know if you can see. Oh, thank you. That's helpful. So I can stay over here so people can see me on Zoom. But we do see, and so this is the odds ratio here, and the size of the dot is the negative log 10 Q ratio. So excitatory and specific enrichment of excitatory inhibitory cell types. Interestingly, we did not see enrichment for glial or for immune related or endothelial cells for this particular analysis. And so interestingly, we do see that this pattern of developmental disconnectivity was we see spatial enrichment across the brain for both schizophrenia and autism-associated genes. So you can see here, but so schizophrenia, tenfold increase. But where we saw the greatest degree of enrichment was actually this is for a postmortem data set of genes that are downregulated in the brain in adults with autism. And so that was a pretty striking enrichment. And so again, to just think about whether these connectivity alterations are clinically relevant, this was a different type of analysis where we did a brain behavior correlation, specifically looking at this age by genotype interactions within the somatomotor network. And here, and then the correlation with the social responsiveness scale, which is a pretty well-established measure of reciprocal social behavior. And we did see this correlational switch. So we see that in kids, there's a positive correlation. A higher SRS score is worse, more pathological. And so we see this positive relationship in the children, and then it switches to this inverse pattern in the adults. So predicting reciprocal social behavior. So just to summarize that little portion, we do see that this functional disconnectivity, this dramatic reconfiguration over puberty, which is mirrored by developmental alterations in dendritic spine density in the mice, and also some evidence for enrichment of synaptic genes in the brain region showing this developmental switch. And then enrichment for autism and schizophrenia genes associated with autism and schizophrenia genes in the broader population, and also specifically within the somatomotor network, predictive of social behavioral alterations. So at least some suggestive evidence that synaptic-related mechanisms may underlie this developmentally mediated functional disconnectivity. I'm actually going to pause for a second if people have questions about the first part of the talk. Yeah, usually.
Switch, we're going hyperconnectivity to hyperconnectivity. What is happening typically in networks during this period of development? Like, are there typical changes in like how these networks are connecting? So like when we talk about hyper versus hypo, what are we coming right now?
Yeah. Great question. And that was because okay, right. So Shula's question is what is happening? So we're seeing this switch and in the 22q deletion cross species. And so Shula's asking what is typically happening within those networks. So we do have some data also that I'll show in a second that about just looking longitudinally within subjects, which I think is probably the best data to show that. But essentially there are shifts, but not to the level, not to the level of, you know, the dramatic level that we're seeing within 22q. So just in terms of, you know, because we are seeing that interactions, that shift is much more dramatic within the 22q deletion than what we're seeing, you know, in terms of typical development. There certainly are network changes that happen over time. And then, I mean, some of the broader changes are in terms of increased segregation between networks as those networks are becoming more refined. But yeah, so essentially, I mean, we're seeing this pretty strong exaggeration of some of those typical changes. Yeah. That is how I would characterize it. Yeah. And Lucina, you have something to add there. I don't know. Yeah.
Well, mainly that we found this exact pattern in like every autism study we've done with like ASD versus TD in children, ASD versus TD in adults. It tends to be hyperconnectivity, and not with the genetic stuff, just, you know, we don't know what's causing it. But it tends to be hyperconnectivity in kids and hypoconnectivity in adults. So it's like some kind of generalized thing that I think is fascinating that you see it specifically with 22q as well. That's amazing.
I'm glad to hear that. Yeah. Okay. Paimon.
Do you think there's specifically changes in synaptic plasticity that may be driving this, or is it sort of synaptogenesis or?
Yeah. Great question. I mean, there seems to be, and whether it's something that's, you know, developmentally regulated in terms of that synaptic, that shift it's a great question. Maybe you have some more thoughts about what is actually going on there. But, you know, we're seeing this, I mean, in the prefrontal cortex where you wouldn't necessarily expect there to be like synaptogenesis occurring at that time period, right? So it's
Always.
There's yeah. So it's a really good question in terms of what is happening, and is there some, you know, I mean, I could hand wave about there being excessive pruning. I mean, that's sort of just a hypothesis, but in terms of, you know, the exact mechanism of what's changing, you know, we don't know. Yeah.
I'm gonna ask, sorry.
Yeah.
Are there efforts to knock out individual genes in the deletion sort of try to understand which of the genes you want?
Yeah, there are. So that is certainly, and actually in Josh Gordon's lab, they did knock out a specific ZDHHCA8, one of the genes in the locus, and then they saw that they did see that that GSK3 beta hyperactivity within that single gene knockout mouse model, and that was subsequently rescued by that antagonist. So there is some evidence that that particular gene, ZDHHC8, is particularly relevant to that phenotype. Yeah, and then there's, other groups are doing, kind of systematic knockouts of specific genes. One of the things that's really complicated about this genomic region is just that there is an incredible amount of like chromatin contact. And there's a lot, I'll talk a little bit more about this, but it just, at least in humans, there is just a lot of interaction with the whole genome. So a lot of regulation of genes, a lot of like trans regulation that does make it complicated. Everything's more complicated than we think. Yeah, so I'm gonna talk a little bit just specifically about, oh yeah. Oh, sorry. Hi.
I asked, when you saw the shift, was it like specific to like either, local connectivity versus like long range connectivity in terms of changing, connectivity state? Because I guess typically it would go from more local connectivity to more long range connectivity as like the brain developed.
No, so the data that I was showing was based on a global fMRI mapping, but then we did look with the seed-based approach, looking within specific networks, and so then we saw a similar pattern. But yeah, so it wasn't that local to long range specifically shifting. And I know that some of the earlier data on that was a little called into question because of the effects of motion on that. Yeah, but I mean, I think there's still like some decent evidence that is occurring. But yeah, great question. It seemed to be more global than that. And so with that, I'm going to talk a little bit, shift gears, talking more specifically about thalamocortical circuitry, which we have particular interest in, as the thalamus is a really critical brain structure for conveying sensory information from the periphery to the cortex. And I know there's other folks here very interested in studying thalamocortical circuitry as well. So these recurrent thalamocortical circuits, we think really represent a fundamental aspect of large-scale brain organization across mammalian species that may be particularly sensitive to network level disturbances. So again, resting state fMRI is an index of the brain's intrinsic functional architecture, reflecting spontaneous fluctuations in neural activity. These thalamocortical circuits also have particular relevance to sleep, because sleep spindles arise from thalamocortical inhibition that's mediated by the thalamic reticular nucleus, this little structure shown here. And so we have found pretty prominent thalamocortical disconnectivity on resting state fMRI. And this is using a seed-based approach where we just place a seed in the thalamus and look at connectivity with the rest of the cortex. And we see, and again, this is in younger folks with the deletion. It's looking at this over connectivity to somatosensory cortical regions. This is just showing this quantified in terms of this pretty large effect over one effect size. But then pertaining to the question of developmental changes. So this was well, again, Charlie did this lovely initial study back in 2019, and then followed it up with a longitudinal study where, again, we're looking at somatomotor thalamocortical connectivity over time. And so again, seeing that, the same pattern longitudinally that what we saw with those two separate groups done cross-sectionally. And so you can see in the controls, there's, it's basically fairly flat. And then we are seeing this, again, this decrease over time. Now, sleep was an area that I was not, I'm not a sleep scientist, particularly. So, but Kathleen O'Hara is an absolutely phenomenal graduate student in the lab, who I think Kathleen may be on Zoom and maybe can answer some of your questions if you have hard questions about sleep. But we had done just some very global studies looking at sleep disturbance associated with reciprocal, just about sleep disturbance in general. First, just showing that sleep disturbance is substantially greater in people with the deletion than controls. This is not surprising. But then within those, within the 22q deletion patients, those that were poor sleepers had significantly higher scores on the social responsiveness scale, that social behavior measure that I just showed you. And so Kathleen convinced me that it would be a good idea to look more into sleep, and I think I agreed with her. So we know that sleep difficulties are exceedingly common in autism across a variety of neuropsychiatric disorders, probably almost all neuropsychiatric disorders, but 50 to 80% of kids with autism have some level of severity of sleep disturbance, and that appears to be related to the level. This is sleep latency, so delay in falling asleep, related to the functional connectivity within that auditory thalamic circuitry, and so we found a similar pattern in our 22q deletion patients, that increased thalamocortical connectivity being associated with increased sleep disturbance. And so this gave us some thought that sleep maybe, well, we're not the first people to have this idea, but that sleep may be a modifiable and transdiagnostic intervention target. Now, of course, sleep has been really traditionally pretty difficult to study with polysomnography. This is what a typical polysomnography setup looks like. Very difficult, especially for kids with sensory sensitivities. And so this was a pretty cool innovation pretty recently. There's a wearable device, the Dream headband. There's some other ones too, but this is the one that we started working with. It's dry electrodes, just a small number of electrodes across the head, and you can do it at home. And so this was our more recent CART pilot project study, gave us the funding to get started on this pilot project, and which then subsequently we got other funding for, which is awesome. So thank you again to CART. And so I mentioned sleep spindles before. So sleep spindles are an EEG signal of thalamocortical inhibition mediated by this thalamic reticular nucleus. And so essentially, they represent a 12 to 16 hertz waveform. It's present during stage 2 and 3 of non-rapid eye movement sleep, and again, reflecting this inhibition of thalamocortical neurons. And there's a lot of evidence that sleep spindles are important for both sensory gating and for memory consolidation. So this was our hypothesis that we've set out to investigate. This is still very much work in progress. So I'll just tell you about some of our preliminary data. But essentially, the hypothesis here is that we have our genes connected to our brain circuitry, and we are studying this at the level of how this may relate to cognition, symptoms, and of course, diagnosis. So our cognitive task, in addition to some broad computerized cognitive task, we're looking at sleep-dependent memory consolidation using a motor sequencing task. And so essentially we have the person do this task before in the morning, consistent time of day. And so essentially what they're doing is just practicing a motor sequence, just tapping as fast as they can, practicing the sequence over and over and over again. It's just sort of a repetitive task. And then the finding in general is that when people, they do the task one day and then they get better on it the next day after a night of sleep. And I'll show you some data on that. But these are just the demographics of our study so far. This is still, so a small sample so far, still very much work in progress. And yeah, about a quarter of these 22q deletion folks have an autism diagnosis, and some of them are also on sleep medication. So this is definitely, there are a variety of confounds. That is something that we are trying our best to deal with, and it's a pretty wide age range. But what we found so far, and again, this is preliminary, but at particular frequencies, we do see that the 22q deletion carriers are exhibiting increased sleep spindle density, which is kind of an interesting finding. But at 12 and 12.5 hertz, this increase in sleep spindle density. We did not see differences in spindle amplitude and duration, just in the density of these sleep spindles. And so, we can, I'd love to hear ideas about the reasons for that. But just to show you what our preliminary data for our sleep-dependent memory consolidation task looks like, This is our controls, our typically developing controls. You can see that, they're getting better at the task over time in the learning phase. Then the test phase happens the next morning, and you can see that they get quite a bit better. Now, the 22q deletion patients, they get a little bit better, but different, not nearly as much. There's a group by phase interaction where they're not, there's a lower magnitude of overnight improvement. And here you can just see this quantified here. And in terms of relationship to spindles, we see that higher average sleep spindle density is associated with worse sleep-dependent memory consolidation in the 22q deletion patients, but actually not in the typically developing controls. We're actually not seeing a significant relationship here, although these are still very small numbers and some outliers. So again, take this with a grain of salt, but we are seeing a pretty strong evidence for an interaction here, despite the small sample size. So there's basically an altered relationship between sleep spindle density and memory consolidation in the 22q deletion. Why that is a great question. So just in the last bit, how much time do I have, by the way? Okay, cool. I think I'm good. All right. So in the last bit, I just want to tell you about this other really super fascinating piece of the puzzle, which is evidence that duplications at that same genomic locus actually may be protective against schizophrenia. And so this is a recent review from Mike Owen. This is a great review, actually, in Nature Reviews Genetics. But essentially, so here's similar to what I showed you before with the odds ratio for schizophrenia, all the way up here for 22q deletion, down here for 22q duplication. So in other words, if you have a 22q duplication, you're significantly less likely to have schizophrenia. When I first saw this, I thought it was, well, that can't be, that must be a fluke, but it's been replicated a couple of times, including in a Han Chinese population. So very interesting finding. So we are very interested in, trying to understand what's going on there. So this was described in, I believe, 2008 or 2009 as a new chromosomal syndrome. It, interestingly, unlike the deletion, which tends to be de novo, the duplication tends to be inherited, and you can have the identical duplication in a parent and a child, and have a parent who has basically a normal phenotype, and have a child that, while they may not have schizophrenia, they do have they may at least have autism or other developmental delays. So there are elevated rates of both the 22q deletion and duplication in non-syndromic simplex autism spectrum disorder. The data that I'm going to be telling you about, these are the demographics. We have actually quite a few more subjects now that we've been able to continue our study, fortunately. But these are just what I'll be telling you about, where we have 107 deletion, 38 duplications, and then age and sex matched controls. You can see that the psychosis is, we're seeing that in the deletion carriers, as you would expect, none in the duplication, but pretty high rates of autism spectrum disorder and ADHD in both of our CNV groups. And so this was work led by Amy Lin, who's a recently graduated wonderful PhD student, neuroscience PhD student. And so what Amy was looking at here was dimensional traits across cognition and behavior in the deletion and duplication relative to controls. Controls are shown here in red. And so basically, so everybody's normalized to the control group. And so anything down here is more pathological. And so interestingly, the duplication subjects had poorer cognition overall than the controls, but the deletion was significantly worse, as you might expect, given the greater deleteriousness of deletions. But in terms of other cognitive domains, verbal memory and processing speed, they were identical. Similarly, social cognition, really not significantly different between groups. And similarly for ASD, autism spectrum traits, were not statistically different between these groups. But we do see statistically different, dimensionally measured positive and negative symptoms of schizophrenia were significantly more impaired, or we're seeing more severe in those with the deletion. So going along with the categorical diagnostic patterns. I'm going to tell you a little bit about some structural MRI, switching from the functional. And so for those of you that are, yeah, of course.
Is it that still versus?
That is a great question. To me, it is not surprising because at the lower levels of those, because you have, basically the scales are going from, 0 is absolutely nothing to, it's hard to be better than controls on some of those measures. And because at the lower levels of those measures, they're pretty non-specific, Exactly. So there are things like, low social motivation, and then, so things that really blend, and on the positive symptom side, things that really blend with social anxiety. So yeah, no, but thank you for that question. We spent a lot of time thinking about the psychometrics of, is there some way to, be, have a superpower? Yeah. So structurally, and for those of you that don't study structural MRI, just to point out a couple of differences in some of the measures I'm going to mention here. Cortical thickness versus surface area. So measures of brain volume are essentially the product of these two indices of cortical thickness and surface area. But we do like to look at them separately because they appear to be driven by distinct genetic mechanisms. They're both highly heritable, but they're not genetically correlated with one another. And they appear to have different underlying neurobiology and distinct developmental trajectories. So surface area, it's really the total area, area of the cortex that's covered in a given region, whereas cortical thickness represents the average thickness of 6 cortical layers within a region. And this is from a science paper from the Enigma Consortium, essentially showing that surface area is impacted by regulation of neural progenitor cell proliferation in the developing cortex, so really very early in development. Whereas cortical thickness appears to be shaped by later developmental processes, possibly synaptic pruning and myelination. And so here we're just looking at a very interesting distinction between, this was again work led by Amy in the lab, between cortical thickness and surface area. So what we're finding is that the duplication has a thinner cortex than controls. Controls are intermediate. Deletion has the thickest cortex, and this particularly in these medial brain regions and somatosensory, we're called heteromodal association cortical regions. The pattern for surface area was the opposite, very widespread reductions in cortical surface area in those the deletion, increases in the duplication. And then when we look at this is just Z-scored to put everything on the same scale in terms of the brain regions showing the greatest effects. Again, for cortical thickness, we're seeing structures like the superior frontal gyrus, middle frontal, the precuneus. For surface area, frontal pole, the cingulate, the medial orbitofrontal cortex, bank of the superior temporal sulcus. So brain regions that are really critically involved in language and social cognitive neural circuitry. So kind of interesting in that regard. Now, that was just a cross-sectional snapshot. So what about neurodevelopmental changes? And we know from some of these beautiful, recent, very large scale studies that there are incredible changes that occur over the lifespan. This is up to birth. We're seeing a lot of changes in overall brain size, but lots of nonlinearity occurring in brain structure and developmental trajectories over time. And so we were interested, and thanks to our longitudinal study, we could look at this in terms of within subject change over time. And this was led by Maria Jobzakowski, who is a wonderful former postdoc in the lab, now has her own lab at Boston Children's. And so it was interesting, and we're looking at here is the controls are shown in green, the 22q deletion in purple, and the duplication in yellow. And so we're seeing this pattern of more protracted cortical thinning in the duplication subjects compared to the, in the deletion subjects compared to the duplication. And the duplication, they actually are statistically not differing in their trajectory from the controls. But on the bottom here, it's just showing the change per year in cortical thickness. And then for surface area, the 22q duplication are actually failing to show what we see as a normative sort of drop in surface area And so they're shown in yellow here. So kind of they're maintaining that increase in cortical surface area. We're seeing some nonlinear changes in the controls. And then again, this pretty steep loss in the deletion subjects. Now, interestingly, when we divide up those 22 deletion subjects with and without who do and don't develop psychotic spectrum symptoms, we found these two distinct periods where there was a significant difference in the trajectory of cortical thickness. So in this early, between the ages of 8 and 12, we're seeing that they have a shallower slope, but then that continues. It's basically a linear decline. And so this is a steeper slope of cortical thinning in the 22q deletion subjects with who develop psychotic spectrum symptomatology. And that actually really mirrors what we see in our clinical high-risk population. So we were pretty interested to see that. Now for surface area, we are also seeing, kind of a similar pattern, but because this confidence interval is quite wide, so we're actually not seeing a significant difference. But we were encouraged by this as it really gives us some ability to pinpoint the period in development when these differences in cortical thickness are arising. So I'm going to just circle back to the genes in this last little bit. So how might these 22q11 genes drive brain and neuropsychiatric phenotypes? We know that this is what we call a genomic hotspot region that's very prone to mistakes in recombination because there's these low copy repeats that flank the region. The typical deletion, which about 85 to 90% of people have the same, roughly the same deletions, 46 protein coding genes. It's pretty big. So everything is more complicated than we would want it to be. But several of these are highly expressed in the brain, as I discussed, involved in early neuronal migration, also involved in orchestrating cell-fate decision in the cortex, as well as microRNAs seem to be particularly, there's several microRNAs, which are small non-coding RNAs that are involved in sort of mechanisms of basic brain development and plasticity, which are particularly interesting. And as I mentioned before, they're both cis effects, so affecting the the specific genes within the locus. So we're going to see a gene dosage effect on those specific genes, but also trans effects that are pretty strong in terms of their ability to regulate genes across the genome. So I just want to, I think this is my last slide, to highlight some work from Jen Forsyth, also a former postdoc who now has her own lab at University of Washington. So Jen, what she looked at here was she looked at the overlap of gene expression in fetal brain with the 22q genes and how they overlap with gene modules implicated in idiopathic schizophrenia and autism. And so what we're seeing is down here, genes within the 22q locus, and then these modules. And this was work done with a method called weighted gene co-expression network analysis. It was actually pioneered here in Dan Geschwin's lab at UCLA and with Steve Horvath. And so what we're seeing is that the pattern of that these 22q genes really cluster within this module that's strongly associated with autism genes, as well as this M8 module or M7, yeah, this module that is associated with both schizophrenia and autism. And when we look developmentally at the developmental trajectory of the expression of these genes, we see a really interesting, some really interesting patterns of developmental changes in gene expression in the brain. So this red line here is birth. And again, this is based on the Allen, or the BrainSpan database. So we're seeing this high prenatal expression of these genes that are in this M1 module here that's specifically associated with autism, with many 22q genes also clustering in that same module, involved in things like chromatin modification, regulation of gene expression, whereas for this M7 module, it's associated with both schizophrenia and autism, again, processes enriched for processes like gene expression. But interestingly, with this module that's uniquely associated with schizophrenia, we see a very different developmental trajectory of gene expression where it peaks postnatally and is involved in processes like synaptic transmission, regulation of synaptic transmission, and learning and memory. Learning and memory is down there underneath that little thing. So So in any case, these 22q11 genes do seem to have broader relevance. And so lastly, just to sum up, hopefully I've convinced you a little bit of the utility of this genetics first approach, where we're seeing that these high penetrance variants can inform disrupted genes and pathways that may be relevant to broader disease susceptibility. We have an emerging model here, at least a hypothesis that deficient early synaptic pruning, a lack of early pruning could be followed by potentially overcompensatory synaptic elimination over puberty, and some evidence that it's from our structural MRI that it's relevant to the clinical phenotype. Sleep, we think, is important, and that's certainly work in progress, where we're seeing this preliminary differences in intrinsic frequency and functional relevance of sleep spindles that may reflect underlying differences in thalamocortical circuitry. We also see that this is a genomic region with mirrored anatomic phenotypes, where we see convergent effects on autism-related traits that, but divergent for psychosis. So we've got a lot more work to do in terms of ongoing studies in our longitudinal cohorts. We also have larger multi-site studies going on, and then in animal and in vitro models that we hope can really guide translation of more neurobiologically informed markers for therapeutic targets, which is, of course, the goal. And so on that last note, I just want to mention some of our work in progress and future directions. So based on our pilot CART sleep study, we got a larger grant from the Simons Foundation, where we're looking at, we're expanding to looking at other autism-linked genetic variants, the 22q duplication, as well as copy nerve variants at the 16p locus. And then we also, in collaboration with Jonathan Sabat, we have a grant from the CIRM, the California Stem Cell Agency, where we are really attempting to look at our within subject, really between our deep phenotypes in human subjects and relating them to cellular phenotypes in organoid models of these disorders. So with that, I just want to thank particularly all the patients and families who participated, of course, all of our funding sources, and again, my awesome lab for doing all the hard work. So I will stop there for questions. Thank you all for being here.